Hello and welcome to QIAGEN's webinar. Thank you for joining us today. My name is Laura and I'm your host for today's webinar on QIAcuity Digital PCR Applications Detection of Mutations. Before we start, let me quickly introduce you to our speaker today. Özlem Karalay studied molecular biology and genetics at the Middle East Technical University in Turkey and obtained her Master of Science degree in Neuroscience from the MPI for Developmental Biology in Tübingen. She received her doctorate in Neuroscience from the Swiss Federal Institute of Technology in Zürich, and she joined QIAGEN after completion of her postdoctoral research at the MPI for Biology of Aging in Cologne. And since then, she has been involved in developing the Digital PCR Assay portfolio, including mutation detection and CNV assays. I'm very happy to have her here today. And with that, I would like to hand over to Özlem to start her presentation.
Thanks. Thank you very much, Laura, for this nice introduction. I would also like to welcome our audience. Thank you all for your interest in our digital PCR webinars. It's my pleasure today to introduce you to our QIAcuity digital PCR platform, and more specifically, today I will be talking about its application for detection of rare mutations. First of all, I will give you a brief introduction and talk about limitations of mutation detection and how the digital PCR technology overcomes these limitations. I will then mostly focus on showing you various examples for quantitation of rare mutations in wide variety of sample types. So before I start, just a brief disclaimer. I would like to remind you that the products that I mentioned in my presentation are not intended for diagnosis, prevention, or treatment of diseases. Okay, without further delay, mutations can be caused by various agents. They can be caused by intrinsic cellular changes, for example, failure of cellular mechanisms such as DNA replication mistakes, or they can be induced by external factors such as exposure to chemicals or radiation. There are different types of mutations: deletions, substitutions, inversions, insertions, and they can lead to permanent changes in the genome sequence, and they can range from a single base pair all the way up to the whole segments in chromosomes. They can be inherited and they can be passed on. So depending on their frequency in the genome, they can be frequently observed or very rarely observed. But irrespective of their frequency within the genome, mutations are widespread throughout the whole human genome. Some mutations lead to beneficial effects. For example, some mutations account for polymorphisms such as hair and eye color or blood type. So they cause natural genetic variation in humans. On the other hand, some mutations have very severe phenotypes and are linked to common and complex diseases. Here, on the left side, you see a list of common diseases in humans. What you see here is some of these diseases are caused by single mutation events, such as point mutations or deletions, and they have varying prevalence in humans. In fact, more than 10,000 human diseases are known or predicted to be caused by single gene mutations. What you also see here is that some of these diseases are caused by multiple complex mutation events, also even on different chromosomes. A very good example for this is breast cancer. So here on the right side, you see the two mutations, BRCA1 and 2, that lead to progression of breast cancer. So these two mutations are located on different chromosomes, chromosome 13 and chromosome 17. And each of these chromosomes themselves have different mutations that lead to different diseases. So in fact, mutations in more than 4,000 genes are identified in the human genome that lead to single gene disorders or that lead to complex diseases such as cancer. There are two major drivers of cancer tumor development. Mutations that lead to activation of oncogenes, such as BRAF, KRAS, or MEK, and mutations that lead to loss of function in tumor suppressors, such as PTEN, or more famously P53. So they all lead to tumor progression. Multiple oncogenes and multiple tumor suppressors are known to be altered across multiple cancer types. For example, mutations in oncogenes EGFR and ERBB2, as well as mutations in P53, tumor suppressor P53, both lead to ovarian and breast cancers. So although we know a lot about the cancer-related genes and mutations, there are still various limitations for profiling of these mutations in especially patient samples. So what are these limitations? Identification of novel mutations in patient samples, as well as identification of residual mutations with very low frequencies in samples are few of these limitations. Some of these limitations, some of these challenges are caused by the quality and quantity of patient samples. There are various sources for patient samples, tissue biopsies such as fresh or frozen biopsies, cell lines obtained from patients, or liquid biopsies can be used to analyze mutations. Each of these different types of samples have different amounts of DNA. They also have different amounts of, for example, contaminants, which might later on affect the quality of DNA extraction. And each of these sample types also have different sample homogeneities. Depending on their source, patient samples can have different heterogeneity. They can contain multiple tumors, and each of these tumors can contain different type of tissues and different type of mutations. So with the increase in heterogeneity of the samples, the frequency of an individual mutation within the sample decreases, making it difficult for its identification. In such cases, higher sensitivity and resolution of detection is needed. Digital PCR platform provides an unmatched sensitivity and precision for detection of such low frequency mutations, especially in comparison to prior technologies such as quantitative PCR. Digital PCR provides a platform for high throughput profiling of mutations. In addition, combined with the NGS results, digital PCR can be used for validation of new mutations or for validation of residual mutations. Here on the right side, you see the two hallmarks of digital PCR technology: random distribution of DNA samples or DNA molecules into partitions, and the absolute quantification of the DNA molecules based on statistical Poisson distribution model. So the end result, the end readout of a digital PCR run is the absolute copies of wild type and mutated sequences within samples. But what is the advantage of partitioning? So partitioning of DNA molecules or DNA samples increases their effective concentration. Imagine there are two targets of interest in a 16 microliter sample volume. So here the concentration is 1 molecule in 8 microliter, 1 in 8. If we aliquot this, or if we divide this sample into 16 equal partitions, each of partition will be 1 microliter. And according to random distribution, according to the random distribution of DNA molecules, each of these two target molecules will go into separate partitions. Thus, we will have 1 molecule of target in 1 microliter of sample, which leads to the 8-fold increase in its effective concentration. Imagine a second scenario where we are looking at 2 target molecules, for example, 2 mutated sequences, which are diluted among a background of wild-type molecules. So here again, we assume we have a concentration of 1 in 8. If we divide this sample again into 16 partitions, what we will have is that some of these mutated sequences will be sharing a partition with some other wild-type molecules. Here, for example, in this case, we will have 1 mutated sequence and 1 wild-type sequence in the same partition, and its effective concentration would be 1 in 1 microliter. So it will lead to, again, 8-fold purification. In some other cases, however, one of these target molecules will be alone in the partition, which leads to complete purification of this target molecule. Thus, by partitioning the target molecule, by partitioning the samples, we will be increasing their effective concentration and decreasing also the effect of interfering molecules. This will allow us to detect much lower mutation frequencies with increased sensitivity and accuracy. QIAGEN offers a digital PCR platform which is optimum for detection of rare mutations. Depending on the sample availability, input volume, as well as the desired sensitivity of mutation detection, 24 well plates with 26,000 partitions or 96 well plates with more than 8,000 partitions can be used. Probe PCR reagents are optimized for use with mutation assays and provide the best performance in nanoplate microfluidic environment. Depending on the number of samples to be analyzed, users can select from either 1, 4, or 8 plate instruments, allowing for high throughput screening of mutations in large number of samples. Results from a digital PCR run can easily be analyzed using QIAcuity software suite, which provides an accurate and detailed analysis of mutations in samples. And important consideration for mutation analysis is actually not just the platform used, but also the assays. QIAcuity mutation assays are LNA-enhanced primers and probes, which provide highest sensitivity and highest discrimination power between wild-type and mutated sequences. 2 mutations can be detected simultaneously using 4 fluorophore combinations. FAM and hex fluorophores for the first mutation, Otto 550, and ROX fluorophores for the second mutation. LNA mutation assays are wet lab validated assays with sensitivity as low as 0.1% mutation frequencies. So from now on, I will be showing you data obtained using QIAcuity LNA mutation assays. So here you see a list of mutations, and these mutations are hotspots, and they occur quite frequently in multiple cancer types, such as EGFR, KRAS, and BRAF. So we generated synthetic templates with varying degrees of mutation frequencies, all the way from 100% all the way down to 0.1% mutation frequencies. And as you can see from our results, LNA mutation assays and various assays listed here are accurately identifying the mutation frequencies in each of these samples. Next, I would like to show you some results that we obtained using FFP samples, formalin-fixed paraffin-embedded samples. So we used Horizon standard templates, which have varying degrees of fragmentation, mild, medium, and severe. And we checked these templates for BRAF V600E mutation. And this mutation is expected to be present at 11% frequency in these samples. As you can see from the results, right bottom corner, independent of the template concentration, whether we put 30 nanogram or 3 nanogram inside a single reaction, we were able to identify the frequency of BRAF mutation in these samples. As you can see at the bottom of the results, we were able to detect 10.7% mutation frequency even in severely fragmented FFPE samples. However, here I would like to pause and remind you that there are several challenges associated with using FFPE samples. Formalin fixation is known to lead cross-linking, degradation or deamination of DNA sequences, and as well as fragmentation. It leads to basically destruction of amplicon sequences that we would like to target. And this all together can lead to inaccurate quantification of mutation frequencies. So what I'm showing you here is actually the same templates that we use, but this time we checked another mutation. We used an LNA mutation assays that target EGFR deletion mutation. And what you can see on the right bottom side is actually depending on the increase in fragmentation, the mutation frequencies that we observed increased, leading to inaccurate quantification of mutation frequencies. So there are different ways to overcome this. Control templates can be used that are also equally fragmented, or users can also always target another assays that are known to be present at a certain mutation frequency in the same samples. So I will be also showing you some results from patient samples. As I already mentioned to you at the beginning of my talk, one of the biggest challenges in detection of mutations in patient samples is, in fact, the quantity of DNA in these samples is most of the time the limiting factor. So what we have here is we have FFP samples obtained from colorectal cancer patients, and we tested these samples again for the BRAF mutation itself. And what we did was we loaded 2 microliter of these patient samples directly into a single well of 26K nanoplate. As you can see, we were able to detect as low as 0.5 and 0.8 copies of mutated BRAF sequences in a microliter of these patient samples. Overall, we were able to detect 3.8 and 6.3% BRAF mutations in these 2 patient samples. And on the upper panel, you again see the exact same data. And at the bottom, what I'm showing you is actually our control samples. As I mentioned, it's always a good idea to include control samples, especially when using FFP samples. And what you see here at the bottom is that we generated some synthetic mixture of templates, and we detected the BRAF V600E mutation. That we use the exact same assays, and you can see that we can detect as low as 0.1% mutation frequencies in these samples. Another point of consideration is the specificity of mutation detection assays. And here I will be showing you the results from 2 different cell lines, MCF7 and SKBR3 cell lines. These are widely studied breast cancer cell lines. And as you know, almost 50% of all breast cancer harbor PIK3CA mutations. And we tested these 2 cell lines for PIK3CA mutations. And what we obtained was that and we actually picked PIK3CA mutations that are only single base pair different. We looked at E545K, G, and A mutations in these two cell lines. And as you can see from our results, we detected around 53% E545K mutation in MCF7 cell lines, but we did not detect all the other mutations. And this is also known in the literature. And in SKBR3 cell lines, we did not detect any of these mutations. So LNA mutation assays are actually able to discriminate a single base pair difference in the target sequences. So we use the same exact PIK3CA mutation assays that I showed in the slide before. And this time we tested a patient sample, FFP breast tumor tissue. And in the sample, we detected E545A mutation at a rate of 16.9% frequency. And we also looked at all the other mutations, and we detected them only some of them at a very residual rate. So for example, we detected, along with the E545A mutation, we were able to detect 0.6% C420R mutation. And again, I would like to show you that you can also use our QIAcuity LNA mutation assays on different variety of templates. Here I'm showing you two different sample types. One sample is obtained from FFP breast tumor tissue, and the other one is obtained from the plasma. And we checked again these two different type of samples for the PIK3CA mutation, and we were able to detect around 15 and 16% mutations of this gene in the two samples. And on the right side, again, you see our control group, our control samples, where we generated synthetic samples with 0.5% and 0.1% mutation frequencies for this particular mutation. And again, the same assays can detect as low as 0.1% mutation frequencies. The last but not the least, I would like to also present you the multiplexing capacity of our platform. So far, I only showed you the data that was obtained by using 2 fluorophores and FAM fluorophore for detecting the mutated sequences, and hex fluorophore for detecting the wild type sequences in green and yellow channels. And here again, on the left side, you see a typical result that I also showed you before. We were able to detect in the synthetic samples around 8.44% mutation frequency for EGFR T790M mutation. So not just duplexing, but the digital PCR platform allows for multiplexing up to 5 fluorophores. So if we would like to detect 2 mutations simultaneously, we can use 4 fluorophores in combination. For the first mutation, we can use FAM and hex fluorophore combinations. And for the second mutation, we can use up to 550 and ROX fluorophore combinations and use green, yellow, orange, and red channels. So here on the right side, you see the results that we obtained when we spiked in a second sample with a second mutation. So as you can see, in addition to EGFR T790M mutation, we also spiked in a sample that carries a 5%, it carries a BRAF V600E mutation at a frequency of 5%. And what we were able to accurately identify was using the FAM and hex combination, the very first mutation, EGFR mutation, at a rate of 8.46%. And the second mutation, the BRAF mutation, we were also able to detect at a highly accurate level at 4.44%. So by combining 4 fluorophores, we will still accurately identify 2 mutations simultaneously. And this brings me almost to the end of my presentation, but I would like to encourage you to go and check our assays in GeneGlobe. LNA mutation assays are all wet lab validated assays, and they cover highly studied cancer mutations, and we will be increasing our product portfolio over time. So this brings me to the end of my presentation. What I told you so far is that I gave you a brief introduction about mutations and limitations of mutation detection, and how digital PCR platform in fact overcomes some of these limitations, and QIAcuity digital PCR platform provides a highly sensitive and accurate quantification of mutations, even in limited amount of samples. And our LNA mutation assays can detect as low as 0.1% mutation frequencies in single wells, and in variety of samples, including, for example, FFPE samples. A large digital PCR LNA mutation assay portfolio allows extensive study of individual cancer genes and also related pathways and related genes. And I would also like to mention here that LNA mutation assays are optimized using QIAcuity Pro PCR kit, and optimized for use in combination with QIAcuity digital PCR instrument. So this brings me to the end of my presentation. I thank you for your attention, and I would be happy to answer your questions.
Thank you, Özlem. Thank you for the very nice detailed information. We indeed already got some questions during your presentation, and yeah, I would say I'm going to start reading them one by one, and you can focus then on answering them. Mm-hmm. So the first questions or the first question we received was about that someone uses digital PCR, and that the main challenge is obtaining enough DNA concentration from plasma. What do you mean concentration-wise? That you use 2 microliter of patient sample in nanogram? Your system detected mutation in DNA samples with concentrations below 5 nanograms. So basically, what is the total input?
So, of course, in combination with LNA mutation assays and in general, the capacity of our QIAcuity platform, we can go as low as 10 to 20 nanograms per reaction volume. For example, for 26K, this would be actually as low as 10 to 20 nanograms in 40 microliters.
Mm-hmm. Okay. Should I go to the? Mm-hmm. Should I? Yeah, I will go to the next one. What is the maximum input? I'm not sure if you have answered that already, but yeah, I will just read it. What is the maximum input in nanogram per well, and what is the maximum resolution per well? Percentage mutant to wild type or fractional abundance?
I'm not so sure if I understand. Can you please repeat, Laura?
What is the maximum input in nanogram per well? Maybe we start with that.
So the optimum range or the maximum amount of DNA that can be added into a single reaction would be 450 nanograms for optimal readout. So between 0.5, and 3 copies per partition would be the optimum range of detection.
Mm-hmm. And is that coming also along with a maximum resolution per well? That was the second part of the question. Resolution per well.
In general, we can detect as low as in terms of nanograms, or I mean, we can detect as low as
sure. It's not written
0.05
here.
copies per microliter.
Okay, good.
Per partition, sorry.
Yeah, I think that will answer it because I don't have also any more information here to the questions. Another one. Do I need a different master mix for doing singleplex, mutation duplex, or mutation fourplex experiments?
No, the same master mix can be used for singleplex and multiplexing experiments.
Okay. And then there was also another question. How much sample can I load to my digital PCR mutation reaction? That's the first one. And then to load as much as possible to find my rare target.
Up to 27 microliters can be loaded actually in 26,000 K plates, number plates.
Mm-hmm. Until which mutation frequency the 8.5 K nanoplate might be usable regarding sensitivity?
When using 8.5 K nanoplates, 96 well plates, I would recommend going as low as 5% mutation frequencies.
Mm-hmm. Then there was also another question regarding nanoplates. Will you design a 26K nanoplate in a 96 well format?
This is a really good question because this is what I was also talking about in my presentation. Sometimes we receive patient samples and these samples are limited in amounts or we don't know the concentration of DNA in these samples, and it would be wise to load as much as possible. And in these cases, basically, we can go for the maximum amount of volume that we can use for the samples and use these plates.
Mm-hmm. Okay. Going through the next one, how many mutations can you detect in one reaction?
2 mutations can be detected simultaneously, although our platform allows for up to 5 fluorophores. We have been developing assays that can detect 2 mutations at the same time using 4 fluorophore combinations, FAM and HEX, and ATO 550, and ROX would be the second mutation.
Thank you. Is the platform open to other assays, non-QIAGEN or non-LNA? Would the results be as good?
Of course, we did intensive testing. Of course, we can use all different type of assays in our platform, but our mutation assays are specifically developed and optimized for our platform. So it would be wiser to use LNA mutation assays in combination with QIAcuity digital PCR platform.
I think we also have received a lot of questions which are not in English. I'm sorry, I cannot I cannot understand them and I cannot ask them. But I saw also a lot of questions regarding if our audience here are getting the presentation. Yes, you will get the presentation. You will also get the recording afterwards. Here was another one. Do you have any CE-IVD assays? That is probably also a good one to ask you, Özlem.
Can you repeat, please?
Do you have any CE IVD assays?
No, not yet, but we will be working on those also soon.
And then what different instruments comprise the QIAcuity systems? Sorry, the QIAcuity system.
I don't understand this question, sorry. Can you repeat?
What different instruments comprise the QIAcuity system? I would understand it the way, yeah.
So we have 3 different type of instruments with the 1 plate, 4 plate, and 8 plates. I think I understand that this is the question. So we have 3 different type of instruments, and depending on the number of samples, depending on the power of high throughput analysis that the users needs, they can go for instruments with higher capacity and 4 plates.
I would like to thank you all for your attention, for all your questions this session. Thank you all. Thank you, Özlem, for you as a speaker here. And with that, I wish you a happy rest of the day. Thank you all.